Hello, and welcome to the Global Immunotalks. My name is Matteo, and on behalf of the organizing committee, it is my great pleasure to introduce my friend Facundo Batista as today's Global Immuno speaker. Facundo is originally from Argentina, but he is the quintessential globetrotter. He received his undergraduate degree from the University of Buenos Aires in Argentina, his PhD from the International School of Advanced Studies in Trieste, Italy, and he did his postdoc at the MRC Molecular Biology Laboratory in Cambridge in the UK. He was then recruited as a group leader at the London Research Institute and was subsequently a tenured member of the Francis Crick Institute and a professor at Imperial College London. Currently, he is the principal investigator and the associate director of the Reagan Institute of MGH, MIT, and Harvard, and the Philip and Susan Reagan Professor of Medicine at Harvard Medical School. Now, Facundo's scientific career has focused on understanding B-cell activation, the fate determination, and antibody production in response to infection and vaccination. He has made several key contributions to the field of B-cell biology, including the establishment of the identity of the B-cell synapse and the mechanism for antigen extraction. He identified key features of the B-cell receptor and the co-receptor organization. He determined the dynamics and the signaling mechanisms that tune BCR function, and he elucidated the key principles that drive B-cell immune responses in vivo. And recently, he has developed a CRISPR-Cas9 mediated strategy to generate knock-in mice expressing specific human immunoglobulin genes. And he has used those to study the immune response to, for instance, HIV and malaria. A couple of things I really like about Facundo is that how he incorporates a lot of different aspects of science in his research, from biochemistry, quantitative biology, the latest technological developments, mathematical modeling. I think this stems from his open and collegial interaction with colleagues and at the institutions he has worked in. And the second thing is the focus he has on mentoring and on the next generation of scientists. When I first met Facundo, I was a postdoc and he was already an established PI. He's much older than I am. and I know he was extremely supportive of me from the very beginning, and I really appreciated this. And now I can witness how he makes an incredible effort, not only in mentoring his lab members, but also the young PIs at Reagan and his capacity as director, and also in the capacity as a journal editor, to bring in young faculties as editorial members to shape the future of science publishing. So I really appreciate that. And for his work, Facundo has received a number of awards, including the Royal Society Wolfson Research Merit, the Embo Young Investigator Award. He is an elected member of the American Academy of Microbiology, the European Molecular Biology Organization, and the Latin American Academy of Sciences. And he's also a fellow of the UK Academy of Medical Science. He served as an editorial board member of Science, and he's currently the chief editor for the Embo Journal. So Facundo, thanks so much for accepting our invitation to speak today. And we look forward to your talk, which is entitled Informing Vaccine Design by Defining the Rules of Antibody Responses. Now, before I hand the floor over to you, it is a tradition of the Global Immunotalks to ask, a personal question to get to know our speakers a little bit better and perhaps to inspire the next generation of scientists. And the question we would like to ask you is, as an immigrant yourself, what advice would you give to a young scientist who is training abroad?
Yeah, first let me thank you, Matteo, and the rest of the organizers for setting this wonderful platform of interaction across the globe. I want to congratulate you with that, and it's a fantastic enterprise. That is a really good question. I think that the answer to that will be really to believe in yourself and to pursue your dreams. In other words, I left Argentina when I was very young, and I was 23, and I kind of didn't speak any Italian or English at all. And for me at that time, it was even difficult to believe that I could be equal as the others. So something I would say, because these days labs are really very cosmopolitan, is that sense of believe in yourself and pursue a dream that you believe and do sacrifice accordingly. I mean, but for me, that was very important. And I wish I could have trust more myself in doing things looking back, but I hope that the young people can do that and the world offer that level of in cosmopolitan and diversity. And so do not feel less. That is my feeling there. Yeah.
Thank you, Facundo. Thank you very much for sharing. So without any further ado, I think you can share your screen and
can you see my screen already or?
Not yet. Let me see. Yes, perfect.
Works. And Mateo, thank you very much for having me. And again, yes, you are much younger than me. And it was always a pleasure to interact with you and from the very, very beginning. So my name is Facundo, as Mateo expressed. This name is very traditional Argentinian, and this comes with a very similar accent that Irina and Carla that organize these meetings. We have this very particular accent that distinguishes Argentinians living abroad. And my talk will be very much focused on B cells. And really, truly, I am an all-time B cell biology. And one joke that was done at the beginning when I was interested in B cells was the notion that B cells were called B because they were boring. However, I think the times for that has changed, and in particular for a couple of reasons that I will elaborate. Reason number one is that probably the best biotechnological discovery in history is without doubt vaccines. This very simple intervention has really saved millions and millions of lives. And it has been a game changer in lifespan, a game changer in our recent history with COVID and what is the effect that these vaccines have to protect. And in particular in the US, that we are a significant statistical data, we can see that some diseases has probably been even eradicated, and hopefully the same happen soon across the globe. But this experiment of vaccine design was born 200 years ago or more, in where Edward Jenner did one experiment today that today will not be allowed. Now that experiment will be the one of inoculating the son of his gardener with some purulent secretion from milk cows, because he noticed that milk cows will not kind of develop disease, they will not develop smallpox. So he reasoned that something they were exposed, he could confer protection to others, and by inoculating the son of his gardener, he showed that he was able to protect it against smallpox. And since then, vaccines has evolved tremendously, but in all honesty, most of vaccines are really of a kind of empirical nature. We don't know, even if we know they were and they protect very well, in many cases, we don't know how. But one very strong correlate of vaccine protection is antibodies, and those antibodies are produced by small cells that are factories of antibodies. They produce thousands of antibodies per second, and those live in our bone marrows. And those are differentiated from initially B cells that they express a membrane immunoglobulin, not a secreted version, but a membrane immunoglobulin that is bound to the cell surface when the B cells are naive. But a very important factor that makes B cells not boring is a notion that since I started my PhD, it was kind of 94, and there were several interventions done by monoclonal antibodies that what was happening, what was causing was the notion that they started to prove extreme protection or benefit in disease. In fact, probably one of the key ones was rituximab, that treating non-Hodgkin lymphomas with this anti-CD20 antibody was a game changer in terms of treating disease. And since then, really, the interest in developing antibodies is such that they constitute a market that is estimated to be in the order of 300 billions, if not $1 trillion now with COVID by 2025. But moreover, a large proportion, if not the majority of the drugs that are in clinical trial are antibodies, because they prove to be effective, but also they prove to be safe and easy to produce. And I'm giving these two topics because this is kind of the backbone of my talk today. I will be talking a little about what we know about vaccines, but also how we can use antibodies as therapeutics. And with that in mind, actually, a lot of the work that I was doing and Mateo was doing also in the past, and he continued to do, was to try to understand B cells from the point of view of how they get activated. And here is where I follow really one of my heroes is Antonio Lanzavecchi. That was one of the first introducing the concept of the two signals for the B cell activation. One is the early signal that is kind of the one that the B cell receptor receive upon interaction with antigen, and we and Matteo and many others contribute to the notion that this is happening really on membranes of macrophages or membranes of follicular dendritic cells in vivo. And then this very early signal that happened milliseconds upon this receptor engage the ligand, the antigen, with a certain affinity, then the B cell receptor do something that is critically important. It's going to extract that antigen, and then it's going to internalize that antigen and direct that antigen to endosomal compartments where the antigen will get proteolytically cleaved, assembled into MAC class II, and then re-exposed on the surface of cells. And this will be the key marker of key specificity for T cells to receive the signal 2. That is separate in time and space. I mean, this signal happen milliseconds, and these signals will happen 10 to 24 hours later, and they are going to happen in different regions of the lymph node. But if B cells receive this signal 1 and this signal 2, what they are going to do is they are going to initially, by day 4 of the immune response, produce these small antibody factories that are extrafollicular plasmablasts, that they are short-lived. They are the ones that just secrete antibodies that are really no high-quality antibodies. These are the antibodies that are encoded by the repertoire, and they are generally low. And the reason why they are effective is because they are in these tentamers, and they have really a high avidity for the antigen. But B cells can do also something rather unique. What is that they are rather unique is that they, after about 5 to 7 days, they are going to form inside the lymph nodes. These structures are known as germinal centers. And what I'm saying by unique is that B cells at this stage are going to start to mutate the region that encodes for a variable domain of the immunoglobulins. And this is at a tremendous cost. We know that mutation on DNA can cause cancer, and this highlights the importance of this process in terms of evolution, why B cells has kept that process, and the reason that they have kept that process at the risk of causing mutations and eventually causing cancer is the one that is the process by which these antibodies can acquire higher affinity. And it's through this process of affinity maturation that high affinity memory cells and plasma cells, that they are going to be long-lived now, not short-lived like these ones here, and they are going to encode for affinities that are much higher in affinity. And this is the process of affinity maturation that we have been very much, as well as many others, trying to understand over the years. But the point that I would like to remind you for the interest of this talk is this notion of what is a germline or a B cell receptor that is genetically encoded, and what is a B cell receptor that has matured and accumulate these many mutations that require many cycles of T cell presentation inside the germinal center. So what my lab does? Well, my lab right now has changed slightly to moving from analyzing this in a quantitative and extensive way, but we kind of focus more now in B cells and antibody responses in vivo. And a way that we do this is by interacting, by asking fundamental questions about this, but trying to really also inform vaccine design. And for that, actually, we use a lot of mouse models in which we take different immunogens, such as HIV, flu, or malaria, and we try to inform how to elaborate immunogens that they will be highly effective, or they will work also in humans. So, and for me, a game changer was my contact with HIV, and one of the reasons why I joined the Reagan Institute, because the Reagan Institute has several PIs based in Durban in South Africa, and we all know what was the impact of COVID-19 into our society. Completely paralyzed the society, completely scared the whole humanity. But let me tell you that something that I wasn't aware, and probably a lot of people is not aware, is that in Durban, in this area of Africa, girls that are 23 to 24-year-old, about 44 or 50% of them, they are already infected with HIV. And although this can be managed through medicines and antiretrovirals and everything, if you really want to have an impact into this, what is absolutely needed is a vaccine design. And what was a game changer for me was the notion that broadly neutralizing antibodies has been identified, that they are able to recognize epitopes on the capsid of GP120, that is the protein that is on the surface of HIV, that are resistant to mutations and they are able to neutralize the virus. And why this is important? Because HIV is a virus that mutates a lot. We are today very scared about how cold is able to escape our immune system, but HIV is a much more difficult target because this is the amount of the total pandemic for flu, the amount of variability that we have in the cohort. This is how much an HIV virus changes in an individual person. This is how is the cohort in 1991 of the whole patients of in HIV in Amsterdam. This is how diverse the different HIVs can be in Congo in 1997. And the concept that I'm trying to say is that while COVID chased somehow and is able to recombine, is able to escape, HIV is much more smarter than that because her rate of mutations is much higher, and therefore escaping to antibody response that will evolve is much more complex. And that is why, for me, a game changer in the field was that was not my work by any means, but the work of many others, such as John Mascola at the VRC, Dennis Barton at Scripps, Michelle Dunstensweiss at Rockefeller, and many others, was identification of these antibodies and these binding sites or sites of the immunoavailability on the virus, which indicates that those antibodies could be triggered, they could neutralize the virus. Therefore, if we elicit those antibodies, we can really kind of have an antibody response that could protect us from infection. So now the problem, as I say, is the mutation, and these antibodies are quite rare. And in this, they are quite rare because, as I said at the beginning, these antibodies are encoded by a V gene or a variable domain on the immunoglobulin that is very heavily mutated. So just to give you an idea, like if you get an infection of flu, you will acquire 5 or 6 mutations or COV in one cycle of germinal centers. These antibodies contain about 25 or 30 different mutations from the germline encoded region of the VCR. But there is also a very important feature that made this vaccine very challenging. And that important feature is really the one of the notion that the protein of the virus is not able to bind, or the B-cell receptor without any mutations is not able to bind at all with the protein of the virus. So the question comes, how you will raise a vaccine if you have a protein that doesn't bind to the B-cell receptor on the surface of the cells to start with? And again, I was not part, but I was fascinated by this concept. The notion is we can then create immunogens that are similar to the mature, but now they bind this with high affinity, and therefore what they do is they induce these naive cells actually to start to acquire mutations in the right directions. And then by using a sequence of those immunogens, we can shepherd the B-cell response and then polish them in terms of getting those protein neutralizing antibodies developing. And for me, beside the notion of vaccine and how important vaccine to HIV is, this constitutes a really fundamental biological problem that is very attractive. That is, can we train B cells really to recognize HIV? Can we train B cells to recognize anything that we want? And this is, in my view, one of the biggest challenges in vaccinology these days, being able to activate in vivo and shepherd those cells in the right direction in order to get these protein neutralizing antibodies that can eventually protect us to HIV. So how do we work about that? And I was very lucky to be part of a large consortium, but also to work with people that I highly respect. I collaborate with a lot of people that I will just mention today, the collaboration that we have with Bill Schiff, my lab, Shane Crotty at Scripps, and Andrew Ward. And in part, some of this work has been published, and other that I will show is not. But essentially, let me tell you through, you can read this because it's published, but essentially these are really unique concept in vaccine design that wasn't developed by me, but was developed by many, and one of the drivers was Bill Schiff. So what people at the Scripps does, this is the work also of Dennis Barton and Brian Brittany there, is they sequence the repertoire, they sequence the naive cells that exist in normal individuals. And why they sequence naive cells? Because they want to identify naive cells that will be precursors, that have the potential to become broadly neutralizing antibodies if they acquire the right mutations. And Bill in particular has done, in this particular paper that we published together, he has done a deep analysis into those cells and identify cells that are not only one individual, but they are present in many individuals. And what he's able to do through in several bioinformatics and generating libraries and then select is, he's able to identify or to produce immunogens that really target mainly the CDR3 of these antibodies, because this CDR3 is very peculiar. I will show it in a second. It's a very long CDR3. So the notion is that he can really target these immunogens to particular CDR3 of several precursors that they have the potential to become broadly neutralizing antibodies. And what we do in the lab is we produce these animal models and we can test many of these immunogens as quickly as possible to move them in the pipeline. And obviously this requires a lot of interaction between labs and collaborators with Shane in terms of the provision of the BCRs that then he clones, to which we use to produce these antibodies. So let me show you a proof of concept here, and let me talk about this B3 glycan N332 that also the talk is going to be referred as HMP or high Manos patch. I mean, the HIV literature is a lot about nomenclature, and I will try to simplify as much as possible this. But essentially, there are this high Manos patch. This is again the protein of the virus, and here we have the high Manos patch that is protein, the antibodies that bind to that, and in particular this one that is VG18. As I mentioned to you, it has a long CDR3 and is able to neutralize many variants of the HIV. And why they require a long CDR3 is because these sites of vulnerability are embedded in the virus because of these glycans that are around. And this antibody has been cloned by Michel Nussensweig and the crystal structure has been resolved by Pamela Bjorkman. But a very intelligent scientist in the laboratory of Bill Stein, very early when this antibody was cloned, he noticed that this CDR3 was very much embedded into these sugars and developed some immunogens that are different to the mature protein that in the field is known as BG505 and 39. These antigens are able to bind particular to the germline version. So they're able to bind to the unmutated version of VG18. So what he did is he produced two immunogens that I will refer by simplicity like N332GT2 or N332GT5. Then what we did is we kind of developed transgenic mice using this CRISPR technology that we use in the lab. And we generate a mouse that expresses particularly this heavy chain. And if we stain the B cells of that mouse using this heavy chain, what we can see is that we stain two colors. We can see that a lot of the cells from those mouse, about 22%, they stain for this GT5 version. And again, if we kind of we sort these cells and sequence, what we see is that all of them express the VG18 heavy chain that is inserted in the right locus, chromosome 12. But essentially, what we see is that we select a large variety of mouse different light chains because, as I said to you, this immunogen mainly binds to this CDR3 and the light chain is just making a strandle there. It doesn't play a major role in function. So the immunogen can target a variety of heavy chains, in this case, one that is transgenic because it's able to bind with high affinity to that particular CDR3. So can we test this in vivo? And here is where I will go through what we do routinely in the lab very slowly, because I will use this experimental concept several times along the talk. So what we have is we create very quickly different versions of this. In particular here, what we have is VG18 germline heavy chain in a mouse background that expresses the CD45.2. This is B220 for visas, but it's CD45.2 marker. And what we can do is out of one mouse, we can transfer very small amounts because in humans, these precursors are found at very low frequency. And what we try is to mimic the situation that we find in humans. So we transfer about few cells in donor mice that are CD45.1. And this particular marker that allow us to identify the CD45.2 will be cells coming from the donor, the CD45.1 are cells, B cells, wild-type cells coming from the host. And this CD45.2 exists in these donors at a very low frequency, that is 7 per million. That is about 10 to 50 cells into the spleen of a mouse. And there is where in those mice we can really immunize them using protein trimers for HIV or nanoparticles, and we can compare how well one works is different to another. And let me take you slowly through the gate strategy. We can initially select this at day 7 and mark for germinal center cells, that we do this as CD95 and CD38. If we look into that particular gate, we can identify the cells coming from the host that are here labeled in blue, that are the CD45.1, and the cells coming from the donor that are these ones here, that are the CD45.2. And we know that because they are CD45.2, they carry the VG18 cell, B-cell receptor. And what we can see is that triggering with these cells, it induces a very robust germinal center, and inside that robust germinal center, we have a large proportion of these cells that not only are recruited, but also they bind the antigen very effectively. So those immunogens in trimer or particles, they work really very well. But what we can do also in the mice is to use these mice as a really state-of-the-art platform. And I will show you some of the things that we can do through in our hands, but also as part of this collaboration. And this is something that many of you may not be familiar, but is MPEM, and this is the first part that is done in mouse sera, and this is the work of Gabe in Andrew Ward Lab, that, as I mentioned, is a very strong collaborator of us. And what they can do that blew my mind away is that they don't do ELISAs any longer. They just take the sera of these animals and they use that sera to bind to them. Through cryo-EM, they can identify whether these antibodies are binding. What we can see is that in mouse that contain VG18 cells, what we can see is that we find antibodies elicited that bind to that particular patch, but also they bind to the base of the trimer, and this is long established that these bases are unprotected. But what is good to see is that these antibodies are able to bind there. And essentially, what we see is that wild types also bind, but if you look carefully, the angle at which they bind is different. And what Andrew can do, that in my view is also, and Gabe, is unprecedented, is they can really resolve this cryo-EM structure at high resolution just from serum of animals. And what you can see is that the animals that carry VG18, they have this long CDR3, they bind to the loop, and they're able to accommodate the sugar, that is a sugar here, N332. However, the antibodies of the mice, they bind in a different way, in a different angle, to this B1 loop here. And essentially, these antibodies, although elicited, they will never become protein neutralizing because they don't have long CDR3s. So whenever we are going to use the second immunogen, we are going to lose them because they will not be able to accommodate a sugar that is protecting that there. So I am blown away by this technology, and Andrew and Gabe are based at the Scripps and has done a lot of analysis for us and for many people in that respect. But also we can go and sequence these cells that we produce, and as I mentioned to you, day 0, these cells express all the heavy chain accommodating different light chains. But at day 42, what we see is that they are selecting particularly for 2 light chains, B kappa that mouse, B kappa 46 and 44, that they seem to be accommodating the immunogen pretty well. What we can measure is, with this single cell sequence, is the degree of somatic mutation. We can see that this immunogen, the N332 GD5, is able to induce high degree of somatic mutation. And most important, that mutation allows to a very broad repertoire. And that is fundamental for the concept of boosting with a second immunogen, because what we want is to generate the diversity of mutations that they will accommodate some of them, and as I show you in a little, they are going to accommodate coming with a second immunogen to bind there. And importantly, if we express these antibodies, and we have done that in many tens of them, what we see is that they gain at least 2 orders of magnitude of affinity to this particular epitope. So in order to summarize and give you a sense of what we do in the lab, we kind of produce, and at the moment we have produced in the order of 200 different lines of knock-in for humans, different B genes, and those relate to HIV, malaria, flu, Zika, etc. And essentially what we have provided is substantial preclinical validation for this N332 GT5 as a good candidate for immunogen design for humans, and this is going to be part of a clinical trial next year. This data was fundamental to kind of allow the GMP product, the go to GMP production of this protein that is going to be tested. But we don't work only in high monospatch, we also work on Apex side, together with Bill and others, and the CD4 binding side, because if we want to have a vaccine to HIV, eliciting antibodies against one epitope alone is not sufficient. What we will need is trimers that are able to germline target different immunogens at the same time, different epitopes at the same time. So we are ongoing work on this, and we have published on the different immunogens. But what I like out of this work, what I show you until now, is that we can prime responses to HIV, and the question is whether we can shepherd them in the right direction. And I will show you in a few slides more whether we can shepherd them or not. Because what I would like to do at this time is really show you a little how the lab works and how we think about things. We are not only testing immunogens, but we are also keen to understand basic immunology by using these immunogens or other noticing. And we were kind of fascinated by the work of a very good friend and colleague, Gabriel Victora, who published a wonderful paper, that a very elegant one, that recapitulates a lot of immunology that has been done in the field, but where Gabriel showed very nicely that each of these is a B cell and is a primary repertoire, a primary GC, and then a primary GC you select high affinity cells, but those cells are going to differentiate to memory. And the question is, when you activate, or when you come with a boost, like when we go and get vaccinated with COV and then we get a second boost, essentially what Gabriel showed is that the vast majority of those cells will not go to the germinal center, but they will go and become plasmablast. So why this is important is because, you know, as soon as you get prime and memory cell different to an IV, we're going to produce high affinity antibodies, and it's going to elevate the antibodies, and this is exactly what we need. But one thing that Gabriel showed, and is something that is established at least for the mouse very well, is that those memory cells really are hesitant to become part of new germinal centers. And instead, Gabriel proposed and showed the notion that is those secondary germinal centers, in other words, germinal centers that happen after immunization for the second time, they are mainly constituted by naive cells. And we were fascinated by this model and very much believing in this model. But one question we want to ask using this model of vaccination, because that is relevant to sequential immunization, is, do we recruit only naive cells? And if we recruit only naive cells, we recruit all naive cells, or some of them may be precluded from this? And in this respect, Gabriel Geron, that he was a PhD student in my lab and a former Gabriel master student here in Harvard, did a very simple experiment. And again, this is taking advantage of this mouse that we do. So Geron, what he did is a simple experiment which he either no vaccinate the mice or prime them with this N332 GD2 protein for HIV. And about 40 days later, or 41 days later, what he does is he adopted transfer naive cells to mouse that were primed or they were not primed. And then 1 day later, he immunized again, so he gave a boost. So the question he wants to ask is, can naive cells really go into the GC? And again, we are using the same strategy as before. We identify donor cells with CD45.1. We identify acceptor cells or BG18 cells from CD45.2. And what Geron saw is that if the mouse are unprimed, he sees after boost that he get a lot of recruitment. However, in mouse that has been primed with this protein, what he observed is that the cells were recruited at very, very low level. Even if they were naive, these are not memory cells. So this surprised us a little, and essentially this prompted us to do a second experiment, that is this dependent on the donor mice, in other words, or the antibodies? In other words, if we use acceptor mice that express now just one B-cell receptor, a B-cell receptor for gene lysozyme, if we prime or not prime those mice, and now we adopted transfer these cells there, can we see recruitment of those cells into the GC? And the answer is yes. You know, in both cases, whether the mice were initially prime or not prime, what Geron sees is that there is a very good recruitment of naive cells to the GC. And this indicates that is something that the cells are producing upon priming, and most likely the antibodies, because this mouse can only produce antibodies to henoglycoside. So to test this, what Jeroen did, and I cut a very long story short, he immunized mice, take the sera, and purify the IgG that then he transferred into other mice. And the day after transferring this, he transferred naive cells and asked the question, is sera from day 7 or sera from day 28 has any effect in the naive cell recruitment of cells? And to cut a long story short, yes, it does. I mean, sera from 7 days, that is a low affinity sera, is germline sera produced for these cells. Do not exclude cells from the GC, but cells that are actually, and sera that is from day 28, that is a very high affinity one, completely abrogate, or not completely abrogate, it diminishes immensely the amount of cells that are recruited. And it's dependent on the amount of antibody that he puts. If he puts little amount of antibody, he gets some inhibition, but as soon as the total amount or the titer of antibody increases, he sees a more substantial inhibition. So what Jeroen is showing here is, look, the antibodies that are produced during the first wave, they have the potential of inhibiting recruitment of naive cells during the second wave. And in order to get convinced out of this, what we did is we tried to prove this in a different experimental system. And for that, we generated mouse that were specific for COV. We generated mouse that are specific for COV in germline, that is, they represent this antibody as a class 4, CR3022, and this mice has very low affinity for COV, or mouse that are very high affinity for COV, and it's the same antibody but with very high affinity. And Jeroen repeat essentially the same experiment, priming or not priming these mice, analyzing cells recruitment. And to cut a very long story short, the results were completely the opposite to the one that he was getting with the HIV immunogen. In other words, the immunization with COV, actually enhance the recruitment of low affinity cells and much more the recruitment of high affinity cells, about 10 times, if not more than 10 times more cells are recruited there. So we scratch our heads a little thinking, why is it that in one case of the COV, this is working to enhancing the response, and why is it in the case of HIV, this is inhibiting the response? And for this, we again use the collaboration with Andrew. And what we study and we understood is that different to what happened to HIV, where I showed you before, the response is very directed to one patch of the protein, and these antibodies are very high affinity and very high titer. The response when Andrew looked in this cryo-EM in sera of humans and in sera of non-human primates immunized with RBDs from COV, what he saw is that the antibody response here is very heterogenic, different to here that seems to be almost monoclonal against one particular patch. And indeed, we couldn't do these experiments with COV in mice because the titer after one immunization is too low, while in the case of HIV, the titers are extremely high. So this raises a question, is the antibodies really blocking that epitope, and therefore I don't recruit cell B cells that bind that particular epitope during the second response? And to test that, we adopted transfer different antibodies. So we adopted transfer antibodies that either bind to this patch or antibodies that bind to all the rest, any other part of the protein. And let me take you slowly through this. But if you take an antibody that binds exactly this patch, what we see is that there is a large inhibition of cells to go into the GC. And this inhibition seems to be dependent on affinity, because if the antibody we use different antibodies with different affinities, we see that the inhibition goes lower and lower. Concentration of these high affinity antibodies is also important, but what we saw is that antibodies that bind to other parts of the protein, they do not have any inhibition whatsoever to cells, BG18 cells that are binding to this part of the protein. So how we can sum this up? Well, what I show you is that we can use preclinical validations models in SARS and COVID-2, And we demonstrate that antibodies elicited during the primary immunoresponse can shape the naive recruitment that we have into GCs during the secondary exposure. And this is fundamentally important because this antibody feedback could be really important to understand how we respond to COVID or any other vaccine. In other words, if our antibodies are really very high titer and very high affinity to start with, we will exclude recruitment of naive cells into a secondary in boosting. However, in the case of COVID, if the titer or we have antibodies binding all over the protein, what we see is that there is an enhancement of antigen specific because we carry more antigen to the follicles, and therefore these immunocomplexes are helping priming more B cells, more naive cells. And again, if we kind of inhibit this, actually, if we have these antibodies that are extremely high, this can work as an inhibitory way by which we will recruit other naive cells, but not naive cells that are binding to that particular epitope there. And this is a little what we tried to illustrate in this cover, that was the cover of November of immunity. We say antibodies can, the germinal center is a little of a party and is a really a castle of protection of the immunoresponse. And antibodies can really help the recruitment of naive cells by forming immunocomplex. But if they are too high affinity, like in this case, and they are at very high titer, what they will do is they will block this response, allowing other responses to other epitope of the protein to occur. So I think that this is really important and is really in terms of understanding. And I will close here. I'm looking at the time, and I will close by saying what are the implications for this in terms of vaccine, or what are the implications for this in terms of sequential immunization. And here is where I would like to refer to a beautiful work that Gabriel Vittora has shown, because he has shown, and this work has been published in Nature, that essentially, if you use different variants, if you use the same variant of COVID, he doesn't get much differential naive cell recruitment. But if he in the middle put an Omicron variant, he starts to get novel naive cell recruitment. And this is very much in line to what we want to happen during sequential vaccination. That is very much in line to what we want to happen in HIV vaccine. In other words, initially, we vaccinate with something that this is the immunogen that has sugars that has been removed to allow B cells to access. But also, as we start to build the sugars in, what we are doing is also blocking high affinity antibodies that have generating and repriming naive cells to allow to acquire more somatic hypermutations. So if we compare antibodies that are generated in the primary immune response against this immunogen GT5, as I mentioned before, they have very high affinity for the immunogen. But if we measure the affinity for these boosting reagents, well, like B16 or B11, we see that they have affinity, but that affinity has decreased substantially. And it has decreased substantially because these immunogens do have more sugars that will make the binding more challenging. Therefore, they will avoid the masking of the high affinity antibodies, because these high affinity antibodies now will have much lower affinities, and therefore they will be able to trigger germinal center cells or memory cells to continue the path of mutation. And this is indeed what happened if we take this boosting reagent, that is a shepherding reagent. So we have mouse prime with GT5, and then one boost now with B16. What we see is that that boost essentially deliver a much higher titer that is specific to B16, that is this boosting reagent. What we see is that this B16 is able to recruit germinal center cells. All of them bind to this B16 boosting, and those cells are in large proportions in germinal centers that are B16 specifics. And just to cut a long story short, actually, we can really do the same with RNA. In other words, we can prime mouse with RNA, and we see with RNA in collaboration with Moderna, this response is much more, is stronger than what we observe with protein, and we can boost this response. And in this case, this response is much stronger of what we have observed with protein. We see that in this case, the prime and boosting regime, all based on RNA, seems to be much stronger in terms of delivering this response into the GCs. So just to summarize until here is that I think that I have provided substantial preclinical validation for this particular immunogen that now is going to the clinic. We have also provided substantial preclinical validation on two potential boostings that are going to, one of them or a related version of this is going to go into a clinic to be tested in this kind of regime in humans. And what I have shown you also is that B-cell responses elicit can be boosted either by protein or an RNA formulation. But in particular, this RNA formulation seems to be superior to the protein. And this could either be because this protein is in a membrane conformation or a membrane expression, it's not any longer a soluble antigen like the protein, or either because the RNA has ways of avoiding this kind of masking of antibodies that we observe here. So the model we are putting right now and we are thinking is that different to protein that need to be carried as immunocomplex, RNA is most likely associated with disease, is dragged into the follicles where they can really activate cells in a very robust way. And very similar during boosting, dendritic cells may play a major role in carrying these immunogens and dragging them to provide a very potent boosting response there. So I am looking at the time and I realized that I was asked to present for 45 minutes. So I probably will jump the story I wanted to tell you into malaria, and just to say only one thing, that is that we can use also these mouse models to improve the affinity of antibodies, and this is particularly relevant in malaria, where the administration of a single dose of an antibody is able to protect people against infection for about 8 months. And just to summarize that and keep in time, I mean, the group of Cedar, this is not our work, has wonderful, wonderful work showing that a single administration of an antibody called CIS43 is able to protect humans from malaria infection for about 8 months. And what we have been working with Bob and Peter Kwon is into producing new variants of these antibodies. And at the moment, we have variants that are at least 20 times more potent than the original one, making the possibility of making this antibody deployed in a large population in Africa, because now we have this antibody at a much higher affinity, at a much higher potency. And the way that we have done that is indeed by producing mice that they are able to be immunized, and variants of this CIS43 antibody are then produced with higher affinity. And with that, I would like to finish. I thought I mentioned the institute, but I would like to finish in time. I would like really to thank a wonderful group of people that are working with me, in particular Jeron, who has been driving the story of antibody inhibition or feedback loop, Shenfei and Ying, who left the lab, who has been driving all this GT5. This has been a wonderful close collaboration with Bill Schaaf and Andrew Wartz, and together with people here at the Rayon, as well as people in Moderna and in Bob Cedar and Peter Kwon at the BRC. And with that, Mario, thank you very much for the opportunity to present. I left the last part open because I felt I didn't want to go over time. So happy to answer any questions or yeah.
Thank you very much, Facundo, for a wonderful talk and a journey through from basic B-cell biology to the design of immunogens. Now, as always, you can ask a question to Facundo via Twitter. You should search for the account Global IMMUNOTALKs, find the tweet that says ask questions for Dr. Facundo Batista here, and you he will reply to that tweet with your question and mention the hashtag Global Immuno, and Facundo will reply directly from the ImmunoSpeaker account. So thanks again, Facundo, and
Thank you, Matteo.
yeah, bye everyone.
Bye.